Oh, thank you very much, Maricelis, and thank you very much for the kind invitation to come here to present our results. So, I'm Charlotte Nellist. I'm from Niab in the UK, and I'm going to talk to you today about our very recent results about the breakdown of Yr15 resistance in the UK. So the company that I work for, Niab, has played a role in mitigating the impact of crop diseases since its inception in 1919. The first disease ratings in the UK for mildew, loose smut and for yellow rust were released in 1952. And in the 1980s, Niab was instrumental in devising inoculated test protocols which still form the basis of today's national and recommended list procedures. So the UK recommended list is a post-statutory testing list of varieties. So in the UK you have to go through 2 years of statutory testing, and then we have this recommended list, which is for sort of the very best varieties, and it gives information to growers on yield, quality, disease resistance, agronomic traits and market options. And the information is completely free. It's released in a booklet which is available online, and also there's an app so you can see all the information about the different varieties. It's funded by levy payers and organised by the Agricultural and Horticultural Development Board. It's updated yearly, and this year in trial we have 38 winter wheat varieties and 12 winter wheat candidates. So the candidates go into trial for 1 or 2 years, and if they're successful then they make it onto the recommended list, and it varies on how long varieties stay on the recommended list. Some have stayed on there a very long time. They have very specific traits that the UK farmers need, so they stay. Others drop in and off depending on perhaps disease resistance. And importantly, there are rust ratings in this, included in this, and just to confuse you compared to other people's results, we score adult plant resistance, the ratings on a scale of 1 to 9, where 9 is resistant, so that's a bit different to what you've been hearing from some of the other presenters. And we also provide seedling resistance ratings as well. And we have a programme for monitoring rust in the UK. So this is the UK Cereal Pathogen Virulence Survey, which I've been involved in for the last 4 years. And it was started in 1967 when we had an outbreak of yellow rust on a previously resistant variety. And since then it's been monitoring many different cereal pathogens, but at the moment we're just focused on yellow rust and brown rust. And until recently we were looking at wheat and barley parody mildew, but unfortunately we don't have the funding to carry on with that work. And the aim of the survey is to identify changes in pathogen races that may have an adverse effect on cereal production in the UK. And we rely on people sending us in samples, so farmers, agronomists, trial operators, breeders and researchers all send us in samples when they see something unusual in the field. So our approach for rusts, we receive samples, we isolate from these samples, and we multiply these isolates, and we put these into long-term storage. And we have a collection dating back to the 1970s, and we have over 4,000 yellow rust isolates, over 800 brown rust isolates, and a small collection of stem rust isolates from the incursion a couple of years ago. We choose about 25 isolates based on the host variety that they've come from, the severity in the field, the location, and we take these for further testing on our differential, in our differential seedling tests, and we classify these isolates into pathotypes. We then have to do some additional spore multiplication to be able to use these on further trials, And we take forward about 5 isolates to do further testing. And we test these on the whole panel of the RL varieties, the recommended list varieties, and the candidates. And this information is used along with observations of young plant resistance susceptibility in the field to produce the young plant ratings in the recommended list booklet. Until recently, we were also doing adult plant trials in the field of these 5 isolates. However, our program's slightly changed now, and unfortunately, we don't have the funding to do those at the moment. And then we choose isolates that most represent the UK population at the time. We provide these to breeders so that they can screen their own material in inoculated trials. We also provide the isolates to the statutory testing inoculated trials and also the recommended list inoculated trials. And between 2019 and 2023, we were able to carry out MARPL genotyping analysis, which Diane has excellently explained earlier. So we've screened 143 isolates during this period, and what we found was that the majority of isolates in the UK at that period belonged to the PstS10, the warrior minus genetic group. And this was about 94% of our isolates. So that's shown by the red colour on the inside of the phylogenetic tree part of the diagram. But within this group, the outside of the diagram is depicting the virulence and the avirulence of the isolates on our differential lines. And so what we see is that there is this broad range of virulence profiles within this PstS10 group that we find within the UK. During this period, we also detected isolates from PstS7, shown by the pink colour on the diagram, and a couple of isolates from PstS8 as well. So this year has been very unusual for us. Normally, yellow rust is first identified in places like Cambridgeshire, where I'm from, Norfolk and Lincolnshire, which is a bit south of the circle that's shown on the diagram. But this year we had the first reports of yellow rust that came from the northeast of England. Oh no, sorry, okay, it's coming. We also had a very warm spring. March on average was 1.3 degrees warmer than usual. In Cambridgeshire, it was extremely dry. I think we went for about 8 weeks without any rain at all. But the northeast did have some rain, and because of these warmer temperatures, we just saw a lot of yellow rust up in this region. So in April, we received many samples from this north region and from varieties rated highly resistant, so from this 8 and 9 classification, and they were also supposed to be resistant at the seedling stage. And then as time passed, the numbers increased, and we did start to see samples in the east of England as usual, but we've still got this heavy presence of lots of samples coming in from the northeast. So we asked the question, has the UK population shifted? This is just some pictures that we received. So we've got this variety KWS Dawsum and Champion. This is a blend of 2 varieties, and they both have or did have resistance at the seedling level. But yet we were getting reports of these high levels of yellow rust observed on them. And 66% of the samples that we received up until the 9th of June were from varieties from this very high classification on our recommended list. Were reported previously to have seedling stage resistance. So that's just shown by the dark green colour on the pie chart. So normally what we see is a lot more samples coming from our lower classification, which is the blue colour, or from susceptible varieties, which is in the grey. So we carried out pathotyping. We took 3 of these very early samples that we received and bulked up spores as quickly as possible, and carried out pathotyping on our differential set, where we observed that there was virulence on YR15, and this came from all three isolates that we tested. So they all confirmed to carry virulence to YR15. And looking back at the data, this isolate, the pathotype that's depicted at the top, this isolate actually came to us in November 2024. So this virulence was in the northeast of England in the autumn and overwintered, and then was able to take advantage of the conditions up there to spread very, very rapidly. So as I mentioned, we're not currently performing MARPL at Niab at the moment, so Diane kindly offered to genotype some of our isolates for us, so we sent 2 isolates to her, and this showed that the isolates sit within this PstS10 clade, and this confirmed that this is likely a mutation in a local European race and not an exotic incursion. So we wanted to look at the UK wheat recommended list varieties to see how many varieties actually possessed YR15. So just a bit of background about YR15. So it was identified, it's a major gene, as has been explained a few times. It provides broad spectrum resistance at all stage resistance, and it was identified in the 1980s. And in the late 1990s, it was introgressed into the European breeding material, and then from there, it's been deployed worldwide. And the first reports of virulence to YR15 was in 2002 from Mogan's lab, and this was a single or 2 isolates that were part of the old European genetic group. And then in 2014 from Pakistan, there was a report of 1 isolate that also carried virulence for YR15, but in this whole time period, they're the only ones, to my knowledge, that have been reported. And so this is extremely rare until now. So there's a group in 2015 that developed some CAS markers to reliably predict YR15 using RNA seq bulk segregant analysis, and they developed these 2 markers, R5 and R8, which sit either side of the gene. And then in 2018, the YR15 was cloned and identified as WTK1, which encoded a tandem kinase, pseudokinase protein. So we wanted to look at the UK germplasm and genotype for YR15. So we used the 2 CAS markers, which segregated our varieties and grouped them into the varieties that we'd seen susceptibility, this breakdown of the seedling stage resistant. They all grouped together, and the 2 markers correlated. We also carried out PCR for the kinase domains. So for kinase 1, you get a 1 kilobase product, and for kinase 2, you get a 2 kilobase product. And so we looked at this for a panel of these varieties, and the data corroborated. But we do see that R5 is a more diagnostic marker than R8 in our material. So going forward, we think we can just screen using this marker. And what we can see in this table, so in the first part, we've got varieties that don't carry any seedling resistance. They don't carry YR15. And then we've got this group in the middle where they all did have seedling resistance. We've received a lot of samples indicated by the numerous pluses in the frequency of samples, and they all possessed YR15. So YR15 is present in many UK varieties. We think that, based on our data so far, it's in at least 33% of the recommended list varieties, and at least 91.7% of the RL varieties resistant at the seedling stage carry this YR15. And the varieties that are confirmed to carry YR15 collectively represent 49% of the total certified seed that's being grown in the UK right now. And this does not account for farm-safe seed. But we can see that the variety that I showed you a picture of earlier, KWS Dawsum, that accounts for 13% of the wheat that's in the fields in the UK at the moment. And then of the candidates that are coming through, 44% of those carry YR15. So this extensive growth of YR15 carrying varieties has put a huge pressure on the fungal population to evolve. And we have an annual trade show, which this year was held in Lincolnshire, called Cereals. And at this trade show, we Niab have plots of the varieties that are untreated. So we have a couple of these varieties, these 3 varieties that all had very high levels of resistance at the adult plant stage, had seedling resistance. And in these untreated plots, the adult plants are absolutely smothered in yellow rust. So probably indicating they did not have any adult plant resistance quantitative genes in there at all. So the UK is not unique. We've heard reports of the distribution of YR15 virulence across Europe, and the IPM Arama project is a European project, and it has the inclusion of the cultivar Maribos, known to carry YR15, included in the sentinel plots. So these are distributed across Europe, and they've started to pick up virulence to YR15. So as of yesterday, Mogan's Watch sent out an email, and it's been found in France, Belgium, Sweden, Denmark, the Czech Republic, and the Netherlands on sentinel plots. So just to summarise then, in the UK we saw unusual recordings of yellow rust in the northeast of England on varieties previously rated highly resistant at the seedling and the adult plant stages. Wheat genotyping confirmed widespread use of YR15 in UK varieties. Isolate pathotyping confirmed virulence on YR15. Genotyping confirmed isolates are genetically similar to European PstS10 lineage rather than an exotic incursion. And virulence to YR15 is distributed across Europe now. So for future work, the collaborators will be doing transcriptome and whole genome sequencing to try and investigate the underlying genetic changes that are associated with this breakdown of resistance. So I'd like to thank all the people that have really worked hard over the last few months to try and get this work pushed through, especially Hugh, who's done all the pathotyping. Tansy helped out with all the genotyping of the wheat varieties. Obviously all the UKCPVS samplers that have sent in samples and recorded things for us, and Diane and her team for doing the marple diagnostics. And thank you very much for giving me the opportunity to come here and present this, and thank you for your attention.